Bonjour à tous, and hello everyone. I will present in English so that more people can understand the presentation. I'm a postdoctoral researcher in Strasbourg. I couldn't make it to Paris yesterday because of some train strikes in Germany, because many trains come from Germany through Strasbourg to Paris. So I have to present from the distance. So I will talk about, so now we will switch to teeth, and I will talk about development of a three-dimensional innovative tool, tooth organoids, for personalized medicine in rare orodental diseases. I wanted to introduce first our institute. So I work at IGBMC, which is an Institute of Genetics and Molecular and Cellular Biology, which is situated in the south of Strasbourg in Ilkirch-Grafenstaden. And so it was founded in 1994. It's a huge research unit, CNRS, INSERM, and Unistra, Strasbourg University, and it has public and private parts. Actually, there are three parts. One that we call IGBMC. So everything is IGBMC, but there is one building for IGBMC, another is CBI, Centre de Biologie Intégrative, and finally, Institut Clinique de la Souris, ICS. And there are four departments in the institute. So I work in the team ERO, which is the team works on pathophysiology of Down syndrome and rare dose-affected diseases causing intellectual disabilities, or autism spectrum disorders and other comorbidities. So I had a presentation recently at the institute, and the people were wondering why do we work on teeth in this team? So the answer is in neural crest stem cells. During development, these cells undergo epithelial mesenchymal transition to give ectomesenchymal stem cells that will give different types of craniofacial tissues. And as you may know, so in Down syndrome, for instance, there are some abnormalities of craniofacial development. So in the group of Professor Agnès Bloch-Dupont, we work on craniofacial and oral developments and their associated abnormalities. There are many dental surgeons in the team. I'm not. I'm a scientist. And in this group, we are particularly interested in orodental genetic disorders, such as amelogenesis imperfecta and dentinogenesis imperfecta or dentin dysplasia. So these are rare diseases that lead to defective enamel formation by ameloblasts. So ameloblasts are cells that will produce the enamel, or defective dentin formation by odontoblasts. Odontoblasts are cells that produce dentin. And so, as you can see, the teeth are different from healthy patients. Agnès Bloch-Dupont is also coordinating the reference center for rare orodental diseases, ORAR. And the team in this, the group developed a targeted DNA sequencing tool, which is called Genodent NGS panel. So NGS for new generation sequencing. It allowed to define some variants in genes involved in odontogenesis to help with diagnostics. But among these variants, there are some variants of unknown significance. So the mutations that do not allow to give the diagnostic to the patients because it's not clear if they are pathogenic or not. So the team started developing, as we cannot work on in vivo models of human development for good reason. So the team started developing in vitro models, for instance, cellular models. So they started by characterizing ameloblast-like cells It's AM1 cell line, derived from ameloblastoma, and odontoblast-like cells that are derived. So we work with human dental pulp stem cells that we differentiate into odontoblast-like cells. By using CRISPR-Cas9 technique, we can obtain mutant ameloblastic and odontoblastic cell lines to mimic the patient-specific mutations. And finally, to study the interaction at the interface of ameloblast and odontoblast, the team started, particularly Isaac Maximiliano Bigueno, with Fadi Gerbaka and Soufiane El Faroussi. They started developing three-dimensional organoids, bilayered organoids, made of ameloblast and odontoblasts. The formation of the organoids was quite successful for murine cells, so for the cells derived from mice. However, as you can maybe notice, the organization of human organoids was less successful, so their stability was less efficient. So that's why they wrote the project 3D Biodent, which obtained the INR funding by ANR. And the goal of the project is to develop three-dimensional models of human tooth development containing patient-specific mutations to allow functional validation in vitro of this VUS, and finally to decrease the diagnostic errors of the patients. And the goal of the project is to develop models at the Bell stage of tooth development. So this is, as you can see from the image on the right. So you have the odontoblast-like cells that are synthesizing dentin, and ameloblast synthesizing animals. So it's a crucial moment in tooth formation. But now I'm inviting you for some time travel. 14 years ago, when I was master student in Paris, in the laboratory working on stem cells. So I was studying the adult bone marrow cells and fetal liver mesenchymal stem cells and comparing the differentiation potential of the cells. And I had some differentiation to do in two-dimensional conditions, monolayer, but also in 3D. So what I noticed is that the cell environment was very important for stem cell differentiation. And that's why I continued by doing a PhD thesis in the field of biomaterials. More particularly, I was working on controlling cellular processes using biomimetic thin films made of some biocompatible polymers. So these thin films, also we call them polyelectrolyte multilayer films or layer-by-layer films. They were discovered around 30 years ago by Gary Deschères here in Strasbourg at Institut Charles Sadron. He still works there. So the principle is to deposit positively and negatively charged polymers. It can be, you can use proteins or even nanoparticles. They will interact using electrostatic interactions. So such films can be made of natural or synthetic polymers. Their stiffness can be tuned by cross-linking, for instance, and we can do biochemical functionalization. These films are usually the most used technique is dipping. So you take a substrate and you dip it into different solutions so that they form these layer-by-layer films. Other techniques such as spraying are being developed too. And depending on the deposition method, so you can obtain either thin films or patterns, or you can also coat the cells. You can obtain freestanding objects, and these films have multiple applications. So we wrote a review article very recently with many teams from all over the world working on these films. So the applications are going from cell signaling studies. That's what I was doing during my PhD thesis. It can be used for drug delivery, even for vaccines, and for tissue engineering. And here we'll be mostly interested in this application, which is tissue engineering. And so after my thesis, where I worked on two-dimensional substrates, I went for a postdoc to Japan to work on three-dimensional tissue engineered constructs to the group of Professor Akashi to work on cell accumulation method. So this method has been developed. This method is to consist in depositing fibronectin and gelatin nanofilms on the cell surface. So you coat individual cells, and then by putting the cells into so we use cell culture inserts. The cell, this nanomatrix will help the cells to organize in 3D. So in the panel G, you can see the fibronectin gelatin coated cells and uncoated cells that disintegrate, the tissue disintegrates. I really like this method because first it allows compact cell packaging and cell-cell contacts. So here you can see some cells in the hydrogels and some cells were deposited by fibronectin gelatin coating method. And the cell-cell contacts are important for tissue maturation and other. So the process is relatively fast and simple. You only need a centrifuge and of course many cells. And you also can vascularize it using, for instance, UVX cells. The size of resulting tissues is well adapted for confocal microscopy. And there is also possibility to vary matrix composition to adapt it according to the cell type. So I think, as you could guess already, I joined the project to bring my expertise in biomaterials to help develop these three-dimensional tissue models of tooth development. And I'm currently working on 3 approaches. So we keep this organoid approach, which is very original. Then I use cell accumulation method. And also during my another postdoc in Strasbourg, I worked on hydrogels and 3-dimensional bioprinting. And so I also have this model. And today I will show you just some preliminary results because I only started in October 2023, so the last year. And yeah, I will show you some first results. So we're trying to figure out which method is the best to mimic the Bell stage of tooth development. So first, about some results on the cell accumulation method. As I mentioned, we differentiate odontoblast-like cells from human dental pulp, stem cells. And here in the images, you can see, so my the first thing to do was to try to coat these cells with fibronectin and gelatin nanomatrix. And then I use fibronectin antibodies to visualize this matrix. And if you compare the upper panel and the panel below, so you can see much more fibronectin. So it means that the coating works for this cell type. You can also see some fibronectin on non-coated cells, which is absolutely normal, as the cells will secrete their matrix. And here, so this is a live dead staining. From the left to the right, you have increasing cell number. So when you put more cells, it gives thicker tissues. And the problem in three-dimensional constructs is that the cells don't really like to be inside, and more cells you put, more you will have of dead cells, what we can see, especially for non-coated cells. But with fibronectin and gelatin coating, the viability is approximately 2 times better. So there are two times less dead cells, even in thicker constructs. So this was really encouraging results. So it looked really promising. However, the one disadvantage is that human dental pulp stem cells, they grow well, but still this is not a cancer cell line. So actually for one microtissue, you need about 500,000 cells, which is quite a lot. So I was wondering if I could do organoids from these cells. So the idea was to do the coating and then to use the method we use for preparing organoids in a low adhesion plate. Yeah, and it requires only 5,000 cells per sample, which is much less than 500,000. So I compared the formation of organoids from the coated and uncoated cells. They look a bit different in the beginning. After, yes, it takes a long time to differentiate. So we can see at day 20, for instance, you can see more compact structures. So this is organoids from human dental pulp stem cells differentiated into odontoblast-like cells. And then, so to study the interaction between odontoblasts and ameloblasts, we add ameloblasts that will form a layer around odontoblast-like cells. And so after 1 week, I can proceed to the analysis. So the first results showed that stability was very nice when the organoids were done with the coated cells. And also you can see some expression of ameloblast related genes. So this is amelogenin here in red, and its expression can be observed at the interface between ameloblast and odontoblasts. So very interesting. And for non-coated cells, yes, like previously, they are less organized. And to finish this presentation, some results on 3D bioprinting. So 3D, bioprinting is association of bioinks. So bioink is usually a hydrogel. For instance, I use a collagen 1 hydrogel, which is mixed with the cells, and then you can print it using extrusion. And the cross-linking allows to obtain something more solid. So you can use UV light or temperature. So for collagen, it's temperature. When you put it at 37 degrees, it will stiffen. We are in the process of buying a 3D printer at the moment, but for preliminary tests, it's absolutely normal to use just a pipette. So you can do hydrogels using a pipette in a multi-well plate. So my idea was to embed odontoblast-like cells into collagen matrix, collagen hydrogel, and then, after differentiating them, to put the ameloblasts on the top to allow the interaction. And so the first results showed good viability of HDPSC inside the hydrogels, and also a good adhesion of ameloblast-like cells on the top. Future work will consist in, so I will continue to work on 3D models to optimize hydrogel culture and evaluate the differentiation of HDPSC in all the approaches. And I'm working with a PhD student who joined the project in November, Efsus. She's mostly working on molecular biology part at the moment, so on transfection and genome editing. And at the end, we plan to join our results to produce three-dimensional models with the variants of unknown significance, so with patient-specific mutations to help with diagnostics. That's all. Thank you very much, and thanks
to you,
Ted Ku, for this opportunity to present this project that I like a lot. Thank you.
Thank you very much.